Hello. We're going to begin our workshop with a discussion of pharmacological considerations when using monoclonal antibodies for the treatment and prevention of infectious diseases. And I'm going to share with you two examples for Ebola and Clostridium difficile. And I'm also going to say a little bit at the end about malaria. Here are my disclosures. So I'm going to start with this question. How long do endogenous antibodies persist in the circulation? The answer to that question depends on the antibody class in consideration. And so here's a table showing the relationship between human antibody class and the range of plasma half-lives reported for that antibody class. And the range of half-lives for antibodies ranges from 2 to 3 days for IgE, up to almost a month, 28 days for IgG, subclasses 1, 2, and 4. What are the main distribution and clearance mechanisms that contribute to the apparent half-life of human antibodies? Well, it's important to remember that antibodies have two main functional domains. The hypervariable regions, VH and VL, represent the antigen binding domain, whereas the constant regions, CH2 and CH3, contain the FC binding domains, and they are also responsible for antibody clearance. When an antibody is created and released from a plasma cell, it can circulate by entering the vascular space. In addition, however, it's important to remember that antibodies can cross out of the vascular space and into the interstitial tissue space through a process called convective extravasation. Once that happens, the antibody can also enter the lymphatic system. And it's important to remember that antibodies can be made within and circulate within the lymphatic system. And that fact may contribute additionally to the pharmacokinetic properties of antibodies. Now, the major clearance mechanism for antibodies is receptor-mediated endocytosis. Antibodies are large proteins, and they can't be easily cleared by traditional drug clearance mechanisms like the liver or the kidneys. Antibodies are taken up into endosomes by first binding to a specific FC receptor on the cell surface, the neonatal FC receptor, indicated in this graph as FcRn. Once inside the endosome, the antibody may be broken down into its constituent amino acids for recycling, or it may return to the cell surface and be released to re-enter the circulation or the lymphatic system. Whether an antibody gets broken down in the endosome or gets released and is recycled is dependent on the binding affinity of the antibody for the neonatal FC receptor. And that is determined by the structure and properties of the FC domain of that antibody. So that being the case, might it be possible to customize antibodies in order to extend their half-life? And the answer to that is yes. This is a figure from an experiment carried out in mice using murine antibodies. Each of these antibodies in the different colors here has a different mutation in the FC binding domain that leads to greater binding affinity for the murine FcRn. And when you compare the half-life or the clearance of these antibodies, in orange is the wild type, which is rather quickly cleared from the circulation over the course of a few days, whereas each of these FC mutations results in a greatly extended half-life and a greatly reduced clearance. So this works in mice. Can it work in humans? And the answer is yes. This is a figure from an experiment published by Godinsky and Rich Koop and colleagues from the Vaccine Research Center at the NIH in 2019, looking at the pharmacokinetic properties in human volunteers of several different broadly neutralizing monoclonal antibodies for HIV. In this figure, the maroon is VRC01, the unmodified human monoclonal antibody, showing its relatively rapid clearance from the plasma with a half-life of about a month. But if you compare that VRC01 unmodified antibody to the VRC01 that now has a leucine-serine mutation in the FC binding domain, VRC01LS in the green at the top of the figure, you can see a greatly increased concentration profile over time, reflecting reduced clearance and a much greater plasma half-life. Interestingly, another related monoclonal antibody, VRC07, with the same LS mutation in blue, did not have nearly as much of a pharmacokinetic benefit, and it has lower concentrations over time and a shorter apparent plasma half-life. Other than this LS mutation, these two antibodies are very similar except for changes in their hypervariable domains. And what this is telling us is that we still have an awful lot to learn about how these FC receptor mutations impact the pharmacokinetic properties of these antibodies. Now, I want to mention that it may be possible to extend the half-life of antibodies not through mutations in the antibody itself, but through long-acting platform delivery. And here is an example of that using a bioerodible implant that was specifically designed for monoclonal antibody delivery that was developed by Eric Apple at Stanford University. This involves a polymer nanoparticle hydrogel that polymerizes after injection under the skin and forms a hydrogel that absorbs water over time. And as it absorbs water, the hydrogel expands, releasing the antibody, but also allowing the hydrogel to be broken down by the body. And the polymer itself is otherwise biologically inert. Now, here is a figure showing what happens when you embed a monoclonal antibody in such a hydrogel polymer. In the black squares, you can see the concentrations of a representative murine monoclonal antibody, in this case, an antibody against ovalbumin, as compared to, in the black squares delivered directly subcutaneously, as compared to the blue squares where the same antibody is injected as part of this bioerodible implant system. And what you can see is that delivering the monoclonal antibody by bioerodible implant increases its apparent half-life from about 4 days to 20 days, about a 5-fold increase in half-life. Now, I want to address this question next. Is it better for a monoclonal antibody to be short-acting or long-acting? And I think it depends upon whether you're using that antibody for treatment or prevention. So let's start with treatment applications. I want to show you some data from a study published in 2019 looking at several different monoclonal antibodies for the treatment of Ebola. And the comparison in this study was the antiviral remdesivir. Now, one of these monoclonal antibody combinations called ZMapp consisted of murine monoclonal antibodies with very short half-lives, about 3 days, as compared to unmodified human monoclonal antibodies that had half-lives in the range of 24 to 28 days. And what you can see in this figure is that the remdesivir and the short-acting monoclonal antibodies, the ZMapp antibodies, were not nearly as effective as the longer-acting unmodified human monoclonal antibodies. Although the remdesivir and ZMapp arms had reduced mortality as compared to the natural history of Ebola, which has a mortality of about 70%, the monoclonal antibody 114 and Regeneron antibody combinations had a much greater survival benefit out to 28 days in this study as compared to remdesivir and ZMapp. Now, the viral load benefit of these monoclonal antibodies is shown in this figure. And what's evident here is that monoclonal antibody 114 and the Regeneron antibody EB3 combination had their greatest impact on viral load in the first few days after administration. If you look beyond day 4, then all 4 of these interventions are associated with reduction or clearance in Ebola at about the same rate. So the major benefit for the human monoclonal antibodies appears to have occurred in the first few days after administration. Now let's look at some prevention applications. Now I want to begin with Clostridium difficile. In this paper published in 2017, looking at a monoclonal antibody called bezlotoxumab for prevention of recurrent Clostridium difficile infection. In this study, several thousand individuals were given standard conventional antibiotic treatment for their Clostridium difficile, but at the same time were given a single infusion of placebo or the monoclonal antibody acotoxumab or this monoclonal antibody bezlotoxumab or a combination of the 2 antibodies. And at the end of 12 weeks, both bezlotoxumab and the bezlotoxumab acotoxumab combination were associated with about a 50% reduction in the risk of recurrence of C. difficile. Acotoxumab alone had no benefit in this study. I want to point out that this is a combination prevention and treatment application because the monoclonal antibody was given at the same time as the onset of antibiotic treatment. But because the antibodies persisted after treatment was discontinued, there could be both a beneficial effect early in treatment and a beneficial effect after treatment for preventing recurrence of infection here. I'm going to finish with the example of malaria. And here is a figure from a study published in 2022 of a monoclonal antibody CIS43 in the LS version. So this is an extended half-life human monoclonal antibody with a half-life of about 2 months. And compared to placebo infusion in the blue, at the end of this 6-month study, both doses of CIS43LS significantly reduced the risk of developing recurrent malaria in a dose-dependent fashion, with the 10 milligram per kilogram dose in the orange and the 40 milligram per kilogram dose in the red. And so in this example, an LS version of a human monoclonal antibody at 2 different doses prevented recurrence of acute malaria in a dose-dependent fashion. So in summary, monoclonal antibodies can be selected or genetically manipulated to fine-tune their pharmacokinetic properties. The desired plasma half-life of a monoclonal antibody may depend on the application for which it is being used. We need to keep in mind the potential impact of anti-drug antibody responses, which will need to be considered during development, and particularly whether a monoclonal antibody is likely to be used only once or multiple times in the same patient. For infectious diseases, preferred monoclonal antibody properties will be different for treatment or prevention applications, and whether the antibody is being used for an acute infection like Ebola, Clostridium difficile, or malaria, versus a chronic infection like HIV, which we'll be hearing about a little later. Future approaches could include long-acting drug delivery platforms like implants. So I want to end now and look forward to the next 3 presentations, and we will have time for discussion at the end of all 4 presentations today. Thank you for your attention.